Introduction. Soil has an abundance of biodiversity. A soil contains tens of thousands of bacterial species and billions of individual cells. Soil microbes evolve to adapt to their environment. They can synthesize biochemicals for bacterial warfare, signaling, or acquiring nutrients. Furthermore, they can break down complex molecules. Bacteria can have antibiotic capabilities. Antibiotics are produced by complex biosynthesis pathways, stepwise chemical reactions catalyzed by enzymes within the cell. They may result from enzyme-catalyzed assembly and modification of amino acids, fatty acids, and sugars. Antibiotics are selectively toxic to bacteria, which implies that either they selectively enter bacterial cells or do not recognize a molecular target in eukaryotic cells. Bacteria may acquire resistance through random mutation in a normal gene or a pre-existing resistance gene from another bacterium, mostly by conjugation. Objective is to obtain a soil sample isolate and to discover identification of antibiotic soil isolate. Methods. First is to collect soil. My partner and I picked soil from my backyard. For our experiment, we used the soil from my lab partner's backyard. Second is to isolate soil bacteria. We did serial dilutions with our soil bacteria up to 10 to the negative 6 dilution. Then transferred each dilution to an agar plate. After we incubated our agar plates, for this experiment, we used the 10 to the negative 4 dilution as it showed the best results of growth. Then we made a grid on our agar plates and transferred our bacterial spores from the 10 to the negative 4 dilution over to the squares. Once we transferred our bacterial spores, we incubated them once again. Third is to check for antibiotic properties. We picked the microorganisms that showed halos around them and transferred them to agar plates. If they showed a clear halo, it was a zone of inhibition, which means it was susceptible to the antimicrobial agent. Lastly, we did 16S rRNA sequencing. After obtaining our isolate, we ran PCR on gel. A process of PCR is the PCR cleanup, which removes primers and chemicals to get purified DNA product. The DNA product splits into two tubes, a forward and reverse, both binding to DNA. The forward will bind to the DNA in the 5' to 3' direction. Meanwhile, the reverse will bind to DNA in the 3' to 5' direction. The tubes are sent out to a company to do Sanger sequencing. We will get two files. First is the forward, and second will be the reverse. With our reverse, we do reverse complement to align our two strands and get a consensus. After we get our reverse complement and merge it with our forward to get our consensus sequence. Lastly, we did a nucleotide blast of our consensus sequence to get our result.